Transfer functions describe the input to output relationship of a biological process. They are most frequently encountered in the context of genetic circuits such as logic gates and environmental sensors. We described a clade of genetic circuits that involve the joining of sensors, processors, and actuators. All of these involve transcriptional inputs or outputs, and it will be useful to mathematically relate these variables. When people speak of characterizing a device, they generally are referring to the experimental measurement of an environmental input in the cell's response. For example, the light sensor from Voigt and coworkers involves a histidine kinase cascade that responds to light and outputs transcription. Controlled amounts of light can be provided using a lamp and a mask, and transcription can be monitored by fusing the output promoter to a reporter gene such as LAC-Z and watching the formation of color. In the figure at right, they placed a graded mask over the bacterial culture, allowing a gradient of light over the bacteria. By measuring the color formation in the cells, they can describe the transfer function relating transcriptional output to incoming light signal. Similarly, a genetic AND gate such as this one would be characterized by a transfer function that maps input transcription to output transcription. Typically, this is done by varying the concentration of an inducer with a known relationship to transcript level, and is monitored by GFP formation as an indirect measure of output transcription. When we characterize an existing device, we find out what it really does. However, when we want to design a device, we need some way of predicting what the circuit will do before we built it. I'll walk you through an example derivation so you see how this works. The transfer function is specific to the inner workings of your genetic circuit and can be derived from the differential equation model. So step 1 in deriving the transfer function is to inspect the diagram and write out differential equations. Here we are considering some inducible promoter. The detailed differential equation model describing the inner workings of any specific inducible promoter would depend on additional factors such as the amount of repressor and any other states of the promoter. However, let's keep this simple for discussion. Let's assume that this simple activator model captures the behavior of the circuit. We say that the GFP protein, or GFP mRNA, changes with time by a production term involving a binding equilibrium and a degradation term first order with respect to GFP. This is exactly what we described with the Shea-Ackers model for a promoter under the control of an activator protein with a constitutive background. Regardless of what you write out for the differential equation, the rest of the analysis is decoupled from the details of the equation. You will make a steady state assumption, set this differential equation equal to 0, and then solve for g. And what you get is an expression for g in the steady state as a function of inducer concentrations and various rate and equilibrium constants. You can thus examine what will happen to g as a function of any of these variables. If you examine g as a function of k, you'd see that it's linearly related to this maximal transcription rate. Similarly, if you examine g as a function of I, you'd see that it follows a normal saturation binding curve. Deriving the transfer function is a simple extension of the modeling we have discussed described by differential equations. It is a useful analysis when you wish to predict how your system will respond to any changes in parameters that you might choose to vary in the wet lab.